Okay, so welcome everybody, and we'll talk about bidirectional communication, and we'll start with signals that come from the periphery that go to the brain, and then on next Tuesday we'll talk about signals that kind of come back from the brain to the immune system, so the afferent versus efferent signaling. Does anybody have any quick questions before we get rolling right now? So this is really one of my favorite lectures because it really starts to put things in perspective and really start to talk about how the immune system communicates. And we'll go through a little bit of a review of innate immunology first, just a few basic things, and then we'll try to understand how these signals use the blood-brain barrier or get past the blood-brain barrier, how they modulate behavioral and physiological responses. We'll talk about a little bit more of the mechanisms of action of IL-1, interleukin-1, which is one of the really premier inflammatory cytokines that has a lot of neuroactivity. We'll talk a little bit about regulation and just show you a little bit after that. You've all seen this before. This is the idea of having two arms of the immune system, the innate and the adaptive. Innate is the primary initial response. It's very quick. It depends on what are called molecular patterns. So it looks for things that it knows is foreign and looks for parts of viruses or parts of bacteria or parts of fungus. And it's a very quick response. But you need it. It's your first line of defense. And this little red line here is showing what would happen in terms of the number of microorganisms if you were lacking the innate immune system only. You would never really get a chance to get to the adaptive, so you'd succumb quickly to this. Now, if you're lacking the adaptive only, so the second phase, the more specific phase, you'll see you mount the initial response, but ultimately, if you can't initiate that second arm of the immune system, you'll succumb to infection, right? So you need both arms working, need them working together, and we sort of need this to resolve inflammatory challenges. So one of the parts of the innate immune system is called complement. It's a very ancient system, and what it is you have these protein complexes that can opsonize. Anyone know what the definition of opsonize is?
Doesn't it make like a cell ready to be phagocytosed?
Yes, but do you know the definition of the word?
No.
It's coat. So opsonize means coat. So the protein complexes coat, surround the cell. There are a couple ways that then what happens is you can have immune complexes bind that, and you see that here in this figure, or what's called the alternative pathway. These protein complex form and actually can punch holes in the bacteria. So this is a really old system, and the reason I bring it up is that we have complement capacity within the brain. So many cells can make these complement components, and we have receptors for complement, and that's something that you should be aware of. So just to kind of go through this, once you get this coating on the cell can be then recognized and picked up. So this bacteria that's coated by complement can be phagocytized and lysed, or you'll be brought into a cell and activate that cell. So like a antigen-presenting cell, for example. These are all just different components of the complement. So they kind of all start coming in together to form this complex. And you'll see some of these. They're described with a C, C1Q, C1R, C1S. And you'll see some of this in some of the neuroimmunology papers. Particularly, I know some of you guys study brain development, and this complement neuron microglia interaction actually helps not only with immune responses, but also brain development and the clearing of unwanted synapses. So if you read any sort of those developmental papers, you'll hear about the complement. So just know it's part of the innate immune system, and it's involved in encoding bacteria. So there are really two main pathways. The alternative pathway, this is going to be when your complement starts binding to the surface of a pathogen and tries to punch holes in it. The classical is going to be not only do you have complement, but you have antibody binding to it. This is going to take a little bit longer, so it's the third act in this complement. Then there's these lectins that you may hear about, mannose binding lectins that bind pathogen surface. So I guess really three pathways of complement. And then, we're going to do three things with complement. We're going to recruit inflammatory cells. We're going to coat those pathogens and have them picked up by phagocytosis. And then we can also have this hole punching within the pathogen cell membrane. So again, it's one of the first lines of defense, and we have some overlap within the brain. Another key component of the innate immune system is the very rapid sensing. And a lot of this comes through what are called pathogen-associated molecular pattern receptors. All right, so they are evolutionarily conserved. You find them in Drosophila. Actually, they're first discovered there. Really important in that first line of defense. And, in the CNS, they're expressed on perivascular macrophages, the microglia, astrocytes, and even some neurons. And this is a list. This list is older. It's from 2009, so it's over 12 years old. But I think there's up to maybe 20 different tolls. But what they look for is just a pattern. And you can have these toll-like receptors on the surface of a cell, or you can have them inside the cell. And the ones on the surface tend to bind parts of bacteria, parasites, and the ones inside the cell seem to bind viral particles. So let's just give you an example. So they can be homodimers or heterodimers. And so you see an example here of a toll-1, toll-2 heterodimer. It's going to be binding lipopeptides, mostly from bacteria. Who expresses them? Monocytes, dendritic cells, mast cells. So these are all myeloid cells, involved in the innate immune response. And then if you contrast that with something like toll-3, which is, a homodimer, is going to look for double-stranded viral RNA. So this is going to be intracellular. It's inside the cell. And once that toll-like receptor binds something, it's a very quick activation, and it drives innate inflammatory pathways. And you could test this very, very simple. You could take a monocyte macrophage microglia. You could put them in culture with something called lipopolysaccharide, which is a bacterial cell wall component. It's going to bind the toll-4 receptor. You can get very quick activation of pro-inflammatory cascade within those cells. So the reason to bring this up is that almost all these receptors, the intercellular ones and the extracellular ones, show up on microglia, right? So again, it supports the idea of microglia being this important immune sensor within the brain. So a full complement of things here in terms of the toll-like receptors. Astrocytes would probably be the cell that has the second most. It has a number of toll-like receptors, has some of the ones that are intercellular, has some of the ones that are extracellular, and it certainly it responds to things like if you had a bacterial infection within the brain. All right, so astrocytes seem to be very important for those type of responses, mostly through this toll pathway. Now, interestingly, neurons also have tolls, not the extracellular ones, but the intercellular ones. And what this sets up is a dynamic where the neurons can respond to viruses that are infecting them. All right, so a neuron will be able to sense to sense a pathogen that's infecting it. And this will become important, especially as we talk about some of the CNS viruses. So I told you that this molecular patterns is an ancient system, all right? And it shows up across evolution. And you can look at, from this slide, you can look at the connectivity to other cytokine pathways, okay? So these toll-like receptors and cytokine receptors actually look a lot alike, especially in their downstream signaling. Now, when I went to college, all you ever heard about was the PKA cyclic AMP kinase pathway. So for you guys right now in neuroimmunology, mostly what you hear about is the NF-kappa B pathway, okay? So let's just take a little closer look at this. I told you about LPS before, gram-negative. It binds Toll-4. There's a co-receptor, CD14. You're going to find this pretty highly expressed on a lot of myeloid cells, including microglia. The downstream activators here, MyD88. These things like are kinases. You guys know what kinases are. Kinases are enzymes that add phosphate groups. They can add it to a serine, threonine, tyrosine residue. So there's serine phosphorylation or tyrosine phosphorylation events. And this changes the conformation of proteins and can allow an active enzymatic site to show up. And then what we see here is the activation of NF-kappa B. And I have a better slide for this. Next slide will show this a little bit better. But what this slide shows you too is that you see a lot of these same intercellular signaling molecules attached to the IL-1 receptor. So you have the MyD88, this is an IRAC, this is a kinase. This is the NF-kappa B NF-kappa B initiating kinase. So they all activate this same kinase. So whether it's a Toll-5 response, a Toll-4 response, an IL-1 response, or even a TNF-alpha response, one of their main signaling mechanisms is NF-kappa B. Okay, so why is that important? So here's my version of that slide, and I think you should know this. I always ask a question about Toll and NF-kappa B. So let's just go through this with this LPS example. You have LPS bind to Toll-4. It requires a cofactor, which is CD14. This interaction will bring these adapter molecules like MyD88 together on the cell surface here or within the plasma membrane. You'll attract these kinases, which generally act as sort of, you'll see these docking proteins. And it'll activate this NF-kappa B initiating kinase, NIC kinase. There's some other names for it, but for our class, we'll call it that. What this does is phosphorylates this I-kappa B. Now, it turns out that NF-kappa B is sequestered to the cytoplasm. You guys probably know this, hopefully, that in order for a transcription factor to be active, it has to be able to get into the nucleus. And what does it need to get into the nucleus?
Nuclear translocation signal.
That's right. And so it turns out oftentimes that you have sequester proteins that bind to transcription factors and it hides that signal. So it effectively cannot get into the nucleus. What this NIC kinase does is it phosphorylates the I-kappa B, which is the inhibitory part. And this phosphorylation, this is a serine threonine phosphorylation, then sends it to be ubiquinated, which is a cell process that we generally use to destroy things. It goes to the proteasome and chops up the inhibitor of NF-kappa B, the I-kappa B. This allows this heterodimer of P65 and P50. Now it reveals that nuclear localization signal, and now it can go into the nucleus. When it goes in the nucleus, it drives a ton of cytokines. So it drives things like IL-6, IL-1, IL-10, and that's that very quick inflammatory response. These inflammatory cytokines can come up within minutes from from the actual RNA to being translated to protein and processed within minutes of a toll activation. Very, very fast. Part of what this NF-kappa B does too is there's a negative feedback. You have I-kappa B produced, and this comes back out and binds up the free P30, P65, P50 and sequesters it again. So kind of an automatic shutoff. And in fact, if you want to know if NF-kappa B is active in your system, you can actually look for RNA of I-kappa B. It'll be highly increased in any sort of inflammatory event. So this is the NF-kappa B pathway, as simple as I can do it for you. And it is basically everything with inflammation that we'll probably read about. It's the main pathway that you see cytokines and these toll-like receptors activate. Okay, so here's one of the slides I showed you, I think, in our first real lecture, this communication between the brain and the immune system, showing you that there is bidirectional cytokine-driven signals and also brain signals using the sympathetic and parasympathetic nervous system and the production of glucocorticoids and catecholamines. All right, and this is what we're going to start to talk about. And we're going to start here with this, what happens in the immune system. The cell gets activated. Let's say it gets activated in to a bacteria through toll binding, and then we have NF-kappa B, and then we have cytokines produced. How do these cytokines signal the brain? And we'll talk about that, and we'll talk about how the brain recapitulates that signal, and ultimately how behavior and physiology are affected. So the brain has an immunostat. It enables it to perceive and represent what's going on in the immune system. And this is driven by cell communication that is associated with cytokine mediated signals. So some of those cytokines that we started talking about, IL-1, TNF-alpha, IL-6, are going to be important here. And ultimately what happens is you're going to change neuronal transmission. All right, you're going to go from a cytokine profile to signaling within the brain to ultimately affecting neurons. And this presents here in the context of inflammation and infection, changes in physiology like increase in body temperature, and behavioral responses like social avoidance or sleep-like behavior. And before I tell you this story, I want to bring to your attention something called the blood-brain barrier, which we've talked about, right? And so if you're saying, Godbout, if cytokines are being produced, how do they act on the brain? Because you told us already that they're large hydrophilic molecules and they can't get past the blood-brain barrier. Okay, so fair, right? Fair question. So I'll tell you how they get past it. Just to jog your memory here, remember that in order for a cell to get into any tissue, this isn't necessarily the brain, but immune cell, a leukocyte here, or this particularly is a neutrophil, which we talked about, needs to be slowed down from the blood. Remember that the blood is like a river system, so it's moving. It needs to be slowed down. It uses selectins and then adheres with adhesion molecules. It follows a path of chemokines, which are like cytokines, but they help recruit cells. And the cell goes into that space. So there's a number of steps. There's rolling adhesion, tight binding, diapedesis, which is a fancy word for getting into the cell, and then migrates to the site of inflammation. So I've told you this as well. So not only do we have this barrier, remember, here's our blood system. Here's sort of a much enhanced perivascular space. It isn't this big, but the reason I used it to be this big is so that I could actually show you cells in there and not a squished up mess. So you basically have to go through 3 layers. You have to go through the endothelium inner basement membrane, the endothelium outer basement membrane, and then the glial limitants. So to get into the brain for a cell or cytokine is not super easy. So let's talk about that for a second. And this is really important when you think about immune brain communication. And so if we want stuff to get into the brain parenchyma, it has to go through all these layers. And there are situations that this happens, right? So we know we have our perivascular macrophages. If a cell is being encouraged to come into the brain or spinal cord, it's going to go inside this space first. So let's give an example. We have some T cells. If you have the proper response and you're producing chemokines and you'd have increased adhesion molecules and selectins, you'll get T cells into this space where they can interact with macrophages. And this is relatively easy to get into perivascular space. There's not much of a barrier for cells getting in here. That's why you see immune cells going in and out of it. To get past the glial limitants, you have to break it down. Okay. And the things that break it down are called matrix metalloproteases. And there's a whole crapload of them. There's probably 24 different matrix metalloproteases, but they all start with this MMP nomenclature. It turns out that the microglia don't really express these. It's those perivascular macrophages that do. All right. So within this interaction here, if you have immune cells coming in for a reason, these macrophages will be activated. They'll dissolve this glial limitin. And then you can get these cells into the brain parenchyma. You can get T cells, you can get macrophages, you can get B cells once you sort of complete this process. So really a two-stage process. First, getting through that blood-brain barrier into the perivascular space, which I'd say is medium difficulty. And then disruption of glial limitins requires a number of enzymes, including these matrix metalloproteases. Probably the most studied are MMP2 and MMP9. And this is relatively hard. Oh, I think I forgot to, no, I did. It's this slide. So I do want you to kind of think about this. We'll see it again, and you'll definitely see it in some of our disease models and our infection models. So you can see immune cells into the brain. You definitely see that they have to cross the blood-brain barrier. They have to get in the perivascular space. And then into the brain parenchyma. And in many, many models, even when you have active recruitment or chemokine signaling, the cells still get trapped in the perivascular space and don't move on from there. It's a two-step process. You need to get through the blood-brain barrier. And then you have to get through the glial limitin. Now some classic studies have shown that if you don't have macrophages in models of EAE, which is the mouse equivalent or mouse modeling of multiple sclerosis, that you don't get T-cell recruitment. So they think that this step is macrophage dependent. Step 2 requires matrix metalloprotease. So if you knock out some of these matrix metalloproteases in these monocyte macrophages, don't get EAE because the cells can't get into the parenchyma. And one final obstacle to remember is even when T cells are in the brain parenchyma, neurons do not like them there and try to kill them. If you get enough of them in there, they're going to overcome that problem. But this final obstacle for a T cell within the brain parenchyma is the neuron who can express fast ligand. You guys remember what fast ligand does to a T cell?
Triggers apoptosis.
Very good, Sam. So agents that increase blood-brain barrier permeability, probably not surprising, things like cytokines, histamine. Those of you who talked about mast cells, remember we talked about histamine, glutamate, prostaglandins, free radicals, all can, so if you're having an inflammatory response, you're going to attract more cells and it's going to be easier to get into because it's going to reduce that blood-brain barrier restriction. Things that seem to tighten up the blood-brain barrier would be steroid hormones like glucocorticoids, which is a sort of classic anti-inflammatory agent, and norepinephrine, which is another, it's part of the sympathetic nervous system, and also part of the thing that attenuates the immune system. So just because something does increase blood-brain permeability doesn't mean that it actually gets in the brain parenchyma. So remember that. It still means likely the cells get confined to the perivascular space. So, why is this process so difficult? Well, first of all, we're talking about a specialized tissue that doesn't have a lot of capacity for repair. It doesn't have a lot of capacity for being replaced. It's very sensitive to inflammation. We saw that with, we see that with neurons, and we see that with oligodendrocytes. Do you remember some of those factors that kill oligos dead? Nitric oxide, TNF alpha, right? So you want to keep the brain in this more homeostatic situation. It turns out, and importantly, that the two extracellular fluids in the brain, the cerebral spinal fluid, CSF, and the interstitial fluid are really the optimum buffers for neurotransmission. If you start putting cells in there and iron and a lot of protein, it disrupts that. And so you're going to see impaired neuronal signaling. Most of the way, if you were to analyze, the whole transcriptome of cells within the brain at a normal baseline level, you'd find that there are a lot of things that try to keep inflammation in check, right? So we try to maintain that anti-inflammatory balance. In fact, I think I showed you that little balance diagram in the previous lecture. So why sometimes when you read these papers done by immunologists, they make it sound like that's the main way immune cells communicate to the brain. Because to them, it makes perfect sense. And that's how cells get into other tissue. But it's not. It's too simple of an explanation. Turns out there are very sophisticated ways to get cells to communicate with the brain. Okay. So if these peripheral immune cells really aren't normally just kind of getting into the brain and dumping cytokines, how do we get these signals? How do we signal the brain when we have a peripheral infection? So this little stick drawing here is, an infection within the lungs, right? Which our brain is eventually going to sense. So how do we get that? Let's say this is a bacterial response. We're going to have, either the very rapid toll response, and we're also going to get some level of antigen presentation and mediate a macrophage Th1 response. Both those are going to drive pro-inflammatory responses that involve key cytokines, the same cytokines that we've talked about throughout the class, IL-6, IL-1, and TNF alpha. These things in general are going to increase membrane permeability and infiltration. One of the things that they can do is increase body temperature. And the reason that we increase body temperature is to slow down the growth of pathogens. And we do see a ton of change in metabolism and energy balance. So we see that the cytokines are driving a change in energy expenditure. And I'll tell you more about why that's important. Negative outcomes of this pro-inflammatory response, you may have heard of septic shock or cytokine storms or chronic inflammation disease. It would all be part of this sort of ongoing kind of toll or type 1 Th1 response. This again is showing you a macrophage who's picked up some bacteria. He can also, or it, I don't know if it's a he, it can also bind to the toll. So, it can bind to bacteria here, part of the innate response, but it can also pick it up by phagocytosis, present it on the MHC class 2 to a helper T cell. In this situation, it's going to be a Th1 T cell, and the main driver of this is going to be interferon gamma, which is a type 1 interferon. This is going to drive what key transcription factor do you think? Isabel, what do you think?
I don't know, P65.
Yeah, NF-kappa B, right?
Yeah.
So it's going to drive NF-kappa B. You're going to see increases in TNF alpha, IL-6, and IL-1. If we break down what these cytokines do, just globally, we see that TNF alpha increases permeability. It's also involved in body temperature. IL-6 does something called the acute phase response, which is from the liver. It's also going to upregulate a number of the complement-associated proteins. IL-1 is going to increase mobilization of immune cells. It's going to increase permeability. It's also going to be driving some of the fever responses. Now, in an immune textbook, these cytokines have a vast effect on all sorts of tissue. Within the liver, you're seeing complement activation. Within the endothelium, you're seeing, the mobilization of neutrophils. In fat and muscle, you can imagine you're breaking them down for energy, okay? And these things are aimed at reducing viral and bacterial replication to drive specific antigen processing and specific immune response. So do any of you neuroscientists know what the hypothalamus is or where it's at?
It's surrounding the third ventricle.
Third ventricle of what organ?
The brain.
That's right. So very simply, the hypothalamus is part of the brain. And usually in immune textbooks, this is the only part of the brain that they talk about. So magically, cytokines only affect the hypothalamus and only affect thermoregulation, which again, designed to decrease the production of viral particles or bacteria. This is a very important slide. I think maybe one of the more important slides that I do. And this really shows you how those immune signals get to the brain. So the first way would be going back to something that we learned in the second lecture, the circumventricular organs. You guys remember those? Those are areas that surround the ventricles that have a less sophisticated blood-brain barrier. So they're more leaky. And so if they're more leaky, more cytokines can get in. Turns out you have a whole bunch of microglia and astrocytes there. So that's represented here. The cytokine in this diagram is IL-1. Let's stick with that one. It's a major one. If you have a leaky or less constructed blood-brain barrier, these IL-1 molecules can get into the brain to act on other cells, right? The other one would be if you have something happening at the endothelial interface. So the blood-brain barrier not only is a barrier, but also it turns out to be a hub for communication. And it's a hub for communication because it's purported to have cytokine transporters that can take these cytokines and transport them across. Cytokine receptors. So we know that the IL-1 receptor is densely expressed on endothelial cells of the blood-brain barrier. And we can also have immune interactions here and actually get, all those cells that get into the perivascular space, they can still signal the brain. Don't have to get into the brain, they can still signal within that perivascular space. And the last one is that there's direct neural connections, which is very fast. So the vagus nerve is part of the parasympathetic nervous system, and it goes out from the brain to almost all major peripheral organs. And it turns out the basal ganglia of this system are chock full of cytokine receptors. So you get a very quick cytokine signal from the vagus into the brainstem. Now, when all these signals get there, they have to be propagated, okay? This little cytokine signal isn't enough to do anything to change behavior or physiology. So you need to amplify it. And this is, I think, where your perivascular macrophages, your microglia, and your astrocytes work together. And ultimately what you see is that there's a recapitulation of this signal. So some of the same cytokines that come in to signal the brain get amplified by microglia, CNS macrophages, maybe in part by astrocytes, and they ultimately work on neuronal targets. And why do they do that? Because neurons have cytokine receptors. Okay, so let's talk about these a little more in detail. This is an example. This is, I think a slide I've already shown you. This is the rat brain. The little lighter gray here are the ventricles, which is we know is where the CSF is being pumped out. Anyone remember the organ that pumps out the CSF?
Choroid plexus.
Choroid plexus, correct. And so within these areas of the circumventricular organs, we still have our microglia and astrocytes that are sensing that environment. They're going to come in contact with things that are high in the bloodstream, and you can get direct activation that way, right? So you get kind of humoral sampling, which is blood sampling by the glia at the CVOs, and that's why the CVOs are important. And then, when the microglia are sort of real close to the ventricles and they get activated, where do they dump their molecules in?
The parenchyma.
Good.
Into the CNS or into the CSF.
Yeah, into the river, man. And then go everywhere, right? In fact, you could detect them. If you had somebody, who had like a bacterial cyst in the brain, you would detect that. Or if you had a other inflammatory event, you can detect cytokines within the CSF. This is the endothelial interface, actually quite complicated. This is adapted from a really great researcher, Ning Quan, who's worked on this modeling of the endothelial cell for a long time. Really the guy that brought IL-1 signaling at the endothelial cell to the forefront. Used to be at OSU, used to give us a lecture, but has moved on to Florida for the nicer weather, I think. Okay, so there's an idea that IL-1 can be transported across. Okay, there's the idea that it signals to the IL-1 receptor, and the endothelial cells in turn respond by making prostaglandins. Maybe they make IL-1 as well. This is the diffusion idea. And then there's also this idea of, if you have recruitment of cells and they get stuck within that perivascular space, you also see that they can signal, produce IL-1, prostaglandins. So any of these four ways can be signaled. So all this sort of systemic inflammation is going to signal not just one of them, but probably all of them. It just depends. Remember, if you had such a high inflammatory event that you're getting cytokines dumped in the bloodstream at high levels, you're going to have some sensing in the CVOs. If it's more specific, you're probably going to be more at that cell communication level where a cell type will be delivering IL-1 directly to the receptor, like here. So other things that happen is this sort of localized inflammation. So if I, injected your leg with like an agent that's going to cause inflammation, there's a couple things that will do that. You can get this localized inflammation. It's actually going to be sensed locally by neurons, and you can get this activation through the vagus nerve. You actually can see injecting something, then seeing areas of the brain light up. This is an example of that cell-mediated idea, where you can see this is actually an example of psychological stress, a chronic stressor that really doesn't drive inflammatory pathways the way like a head injury would. But you can see that you have these green cells, which turn out to be peripheral monocytes that come in to the brain vasculature. So the red here are the microglia. Maybe you can see their processes. The blue is the vasculature, and the green are these monocytes. And you can see them all sort of stuck in that perivascular space. And it turns out they're providing signals that the brain respond to, and the signal that they're giving is actually IL-1. Okay. We also know there's some cell-mediated surveying, and this is also something that we see with the lymphocytes, the T and B cells. So this is this is work by Yoni Kipnis's lab. If you want to learn more about T cells and the glial or the lymphatic system of the brain, he's probably a good person to read his work. I think he just recently published a big paper in Cell. I think it came out yesterday. We'll definitely take a look at that. Maybe do that for one of our class discussions. But he's shown in his work that there's a normal sort of T cell surveying within the meninges. These T cells actually help with normal brain development and actually help with normal cognitive function. And that when you start impairing the T cells or change the T cell profile, you get impaired cognitive function. So there's a couple different ways we see this cell-mediated responses. It's not always, none of these things are necessarily negative. Inflammation isn't always bad. This is the direct neuronal signaling. This is the parasympathetic nervous system's vagus. The vagus here comes off. It's like one of the cranial nerves, and it comes and you'll see it go to all these important tissues. The fascinating part about where the vagus innervates is that it's often innervating close to these very strong tissue macrophage pools. So in the lungs, you have the alveolar macrophages. In the liver, you have the Kumpfner cells. In the gut, you have the GALT and the sort of gut-associated macrophages. So all the important peripheral tissues that you worry about and that you're sensing, the vagus innervates. And so very small amounts of changes in that macrophage profile. So if they're being activated, they're making cytokine, it's a very rapid communication back up to the brainstem. Not only does it go to the brainstem, you'll see it actually kind of the signal goes around the ventricles too. And if you disrupt the vagus, you can cut the vagus, which is sort of barbaric, or you can paralyze it. And both of those things will attenuate sort of immune brain signaling. It's not the only pathway. Remember, all these pathways likely work in concert. You have neuronal pathways, you have endothelial pathways, you have cell-mediated, and they all kind of work in concert. Now, what's interesting is that you can see direct neuronal activation during infection. So this is an immune challenge with a little polysaccharide. This is this is a peripheral injection, and within 4 hours, all these black cells here are activated neurons within the CVOs. So it's a very quick signal. You also see it in other areas. The NTS is part of the brainstem. All right, so any infection, it's going to be picked up by that vagus nerve system, transmitted, and get very active or very quick neuronal activation, which is likely where you're getting the change in body temperature and some of the probably initial behavior responses.
I have a question.
Sure.
Are those neurons like within the brain itself? Do they actually express IL-1R? Or do you think it's a different signal mediating their activation? So like the activation of the vagus is mediated via IL-1 receptor. But then within the brain, did those neurons actually express IL-1R1? Or
it's probably not. That pathway is not going to be IL-1 driven. It's going to be neurotransmitter driven. So do you know what the vagus uses? Anyone know?
Isn't it like epinephrine or norepinephrine?
Serotonin.
Acetylcholine?
Yes. The last person was right. Okay.
Thank you.
Oh, you're welcome. Thank you for your correct answer. Okay, so I think this slide is really fascinating. So what does all this mean? Why do we even have this communication? And remember, I told you about the change in, how am I doing on time? Okay, I think I'm talking a little too slow, but that's okay. We don't have any agenda here. So let's go to a mouse model. Let's say we give our inflammatory challenge, and we stick to a bacteria challenge here. We're going to get the mice are going to be sick, right? And so it starts off where you injected it. So the macrophages are going to sense it. They're going to sense it, through the tolls. They're going to sense it through the Th1 response with the T cells. They're going to produce cytokines that get to the brain in all the ways I just showed you, right? Once that is activated, we see some physiological changes. So we see the drive to increase body temperature. That's going to be mostly hypothalamic response. Another thing we see is a drive to reduce plasma iron concentration. So that's centrally driven to sequester iron. These things are both aimed to inhibit the growth of pathogens. Now, part of the energy balance is, you think about it, the body tries to conserve energy in some ways and expend them in other ways. So one thing we know is that we see an increase in sleep. I don't know if any of you guys attended the neuroscience lecture yesterday, but the speaker, Ashley and Josie, talked about IL-1 signaling and driving neurons in sleep. So there's direct evidence for that. We also see reduction in appetite, activity, and something that's called increase in something that's called social withdrawal. Now, these are all part of the innate immune response. So happen pretty fast and are sort of doing it based on patterns. And the idea is that these things not only change energy balance, but also do it so that you give the body time to start the adaptive. So you're kind of, it's sort of a time gag. And then when those two systems come together, the adaptive and the innate, you clear the pathogen. And so now your mouse, who was happily eating his mouse food, he's sick here, so he's not grooming, he's not eating, he's kind of alone. And magically, within a few days, 2 days or so, he's back to eating. And it's definitely not the same picture of the same mouse that we started with, right? It is the same picture, but they do go back to normal. And so what's really interesting in terms from an evolutionary perspective, if you start to mess up these pathways, okay, and when we're sick, we don't like to eat, right? And if you start to force feed an animal with a bacterial infection, the odds of increased mortality go up. Okay, so there's a reason why we're not eating. And I guess it's really tied to that energy balance and metabolism. So if you force feed, these are force feeding mice through a tube who have a active listeria infection, the mortality goes way, way up. Okay, this is another thing in terms of behavior. When you're sick and you don't want to be around people and you kind of stay home, you don't go to class, and you sleep on the couch, maybe watching reruns of Law and Order. So that is controlled by cytokines, and it's happening within your brain. So here's a mouse here who will normally engage in the social behavior with a younger juvenile mouse. When he's sick, you'll see he'll pay him no attention. And we can induce this behavior a number of different ways. Let me get my face out of the way. Oh, I guess I can't. We can induce this in a number of different ways. So we can inject IL-1 just into the bloodstream. And we can see, this is the green line, we can see that the mice reduce their social behavior within 2 hours. And that slowly they start to interact more and more. Within 24 hours, they kind of go back to normal. All right, we can also give the lipopolysaccharide injection. It makes them sick, they don't interact, and by 24 hours, they come back. And then we can also give the IL-1 injection, ICV. Anyone know what that means? If I'm injecting something ICV, where am I putting it?
Into the ventricles.
That's right. IP means what?
Intraperitoneal.
Right, so I'm putting it not into their gut, but into their abdomen. So you can see that really one of the main drivers of this behavior is IL-1. This is work done by Robert Danzer, really one of the classic psychoneuroimmunologists out there. He was in France for many years, leading their institute, now is at MD Anderson in Texas. A very interesting guy to talk to, probably one of the brightest guys I've ever met. But showing here that he's giving LPS injections to mice, sees that they get sick. And if he does a vagotomy, which is cutting the vagus nerve, that you totally block this effect. So one of the ideas is that, that the vagus is sensing some of these inflammatory signals in the periphery, sending that quick signal to the brain, and it's part of what's changing behavior. Fever isn't always stopped by the vagotomy. So this is an interesting concept and sort of some classic studies of these behavioral responses to cytokines. Is it this idea of internal weakness, this medical interpretation, or is it more complex? So these cytokines, as I told you, will drive this reorganization of host priorities. Now, is it basically this medical interpretation is all or nothing, meaning cytokines are there, you're going to get sick and you're going to change behavior? Or is it more complex? Is it more of a motivational state? So do cytokines change the motivational state that then you take part, you think about the environmental contingencies, and then behavior changes? So a much more complex view of what might be happening. So Danzer in his lab did this, which I think is a fascinating experiment. These terrible drawings here are done by me. This is a rat. And what they're doing here is these are rats who have a cohort of babies. And it turns out that a rat will, if you disrupt this nest, either move the mice away or disrupt the nest, that this lactating mom will do everything it can to rebuild the nest. So when they give, they're giving these LPS injections. Remember, this is going to be our innate immune stimulator. They did this at 2 different temperatures, 24 degrees Celsius, which I think is about room temperature, and they did it at 6 degrees when it's cold. And so they made the mom sick, pretty sick with this LPS injection, and then they displaced the pups and took away the nest. And so they wanted to see what the mother would do. And it turns out that if it was warm enough for her offspring to survive, she would just bring them back and sit with them again. So she would, no matter where they were in the cage, she'd bring them back, retrieves her things. But because it wasn't cold, she didn't use energy to rebuild the nest. Now, if it was cold, not only did she retrieve her pups, she also rebuilt the nest. So she was, looking at the environment to determine what her behavior was, which I think is fascinating, right? I mean, it'd be the same. Let me go back. It'd be the same thing, right? If you're sick at home watching those Law and Order reruns, right? And there's a knock at the door. Do you expend the energy to get out, to see who's at the door? Probably not. But if you smelled a fire or something like that, you would definitely, you'd run up, grab your kids, and get out of the house, right? So we can, while cytokines change the motivational state, we also can weigh in those different environmental contingencies to change behavior, which I think is fascinating. Okay, so what do these cytokines do within the brain? Here are those 2 big ones that we've talked about previously, IL-1 and IL-6. So during an inflammatory response, they're going to drive sleep, fever, acute phase responses. That's going to be part of that complement cascade. Immune activation. It's going to activate the sympathetic nervous system and what's called the HPA axis, which is the hypothalamic pituitary adrenal gland axis. And we're just going to do these activations at the expense of sex drive, appetite, mood, growth pathways. So the HPG and HPT are growth axes. So you're going to slow growth responses, save energy for these immune responses. Does that make sense to everybody? So it's this balance again. Okay. So really important, I think, in terms of critical concepts of some of the slides I just went through. So the brain represents what's happening in the peripheral immune system. This is cytokine based and sort of represented in this sickness behavior. Sickness behavior is a reorganization of the host priorities, really tied to energy balance. You are mobilizing energy to fight infection, but you're conserving it behaviorally. One of the ideas with this social isolation behavior is that you are limiting the spread of infections. For social animals, like mice and humans, if you're very sick, and we're like we're finding out with COVID, right? And then you go to a big block party, you can get everyone else sick, right? So one of our intrinsic things that cytokines do in our brain are tell us that we're sick and need to stay home and isolate. So that kind of loss of motivation is important for limiting the spread of infection. And if you think about it, what do they do when people come in that are very sick and contagious? They isolate them, right? So it's actually part of our intrinsic behavior. So these behaviors are, lack of motivation, you're going to have increased lethargy, reduction in food intake, increased sleep, and social withdrawal. So what's really cool about this response too is that all of us have been sick before, and it doesn't result in loss of neurons, right? So we can tolerate this level of immune-brain communication. It's reversible. It's evolutionarily conserved. I think it's conserved all the way down to cockroaches, which is surprising, but almost everything that sort of has an innate capacity will have a sickness behavior response. If you disrupt these pathways, so I showed you the force feeding, but also if you start to interfere with the ability of an animal to mount a fever response, you also increase mortality. And you can have all this without having the breakdown on the blood-brain barrier. You don't need infiltration of T cells and B cells, and you don't kill neurons. So our body can tolerate this level of inflammation. It causes a physiological and behavioral response that's beneficial to the host organism. And I just want to say that this concept here is something I think is probably the most important part of neuroimmunology, is that knowing that those signals, the cytokine signals, are tolerated and change behavior in a positive way, right? So if you're immunologist, you go back to that model of just the cells get in the brain and do stuff. That's not right. Okay. This is more work by Kipnis showing, that you have surveying T cells modulating cognitive function, basically showing you that with an infection, this is going to be altered, and you can see altered cognition as well. That's part of this T cell monocyte macrophage loop. So what's important about this is, how many of you guys have ever taken a test when you felt really sick? And how'd you do on that test? Probably not as well as you would have been if you were healthy. So this is something that we see, is that we also have some, these cytokine pathways and disruption in T cells affects memory and cognition. Okay, so how are these cytokine signals propagated and interpreted in the CNS? This is a complex slide, but I want to think about the sort of the time and spatial activation. And speaking of time, how am I doing? I guess I hit my hour. You think I should stop, or you guys want me to finish? Should we do a poll? You gonna make me guess what I should do?
I mean, I guess it depends on how involved the rest of the slides are.
Well, let's do it this way. Let's finish this slide. We'll stop here, and then whatever's left over, we'll start with on Tuesday. Fair enough? Okay. So we're going back to our CNS sensory inputs from the vagus nerve. Okay, so let's say that our vagus is sensing some macrophage activity within the lungs, right? It's going to quickly send that signal to the brainstem. From the brainstem, that signal goes to the paraventricular nucleus of the hypothalamus. And that's going to be one of the areas important for thermoregulation. So this vagus is going to go a couple different areas, and this is really going to be the start of that signal. It's going to happen relatively fast. Now, we know within the blood-brain barrier and those endothelial cells, we have those CNS macrophages. This is when they turn out to be important, those sensing cells. Also, we have those microglia in the circumventricular organs. So these are probably the next signals to come in. That's represented here in orange, number 2. And you're going to see sort of the amplification of IL-1 signaling. As we mentioned before, some of this IL-1 signal is then dropped into the CSF, where it can essentially go anywhere. The third activation state, this is when you're going to get the perivascular macrophages producing cytokines, okay? And 4, finally, you get some of that parenchymal microglia activity. So it takes a while to get to them. And once that gets to them, the signals that come to them could be prostaglandins, it could be IL-1, it could be nitric oxide. This is considered that secondary cytokine phase. Okay, and then collectively, the end target is always going to be neurons. So not only do neurons have cytokine receptors, and I should tell you that the distribution and density of cytokine receptors is very region dependent. So we could talk a little bit more about that. But neurons also respond to prostaglandins, which are from the breakdown of arachidonic acid into these neuroactive molecules, and also nitric oxide. So all 3 of these can influence neurotransmission. So we'll stop here, and we will continue on Tuesday. We'll start off with this slide, I think, again, and then I'll just move some of these slides to the next part, and then we'll go from the brain back out to the immune system. So let me stop recording.